Good afternoon, everyone. Welcome to today's Food for Thought seminar. I'm Liam Blacklock from the Food for Thought team. Some housekeeping information to start with. This is a Teams live event, so you won't be able to unmute yourself or come on camera, but you can pop any questions you might have in the little Q&A section on the side, and we'll cover those at the end. We are always looking for recommendations of speakers to invite, so if you have any suggestions, then please let us know at our email address, which is foodforthought@food.gov.uk. Now, it's my pleasure to introduce today's speaker, Dr. Sarah O'Brien. Sarah qualified in medicine in 1986 at Newcastle University and undertook higher specialist training in public health medicine in Oxford and Newcastle upon Tyne. She has held consultant positions in health protection in Birmingham, Glasgow and London before joining the University of Manchester in 2004 as Professor of Health Sciences and Epidemiology. She moved to the University of Liverpool in 2011 as Professor of Infection Epidemiology and Zoonosis, where she was also Director of the first NIHR Health Protection Research Unit in gastrointestinal infections. She joined Newcastle University in August 2019. Sarah's research covers a wide range of gastrointestinal infections, including foodborne disease. Her research portfolio totals over £45 million to date, with funding from the FSA, MRC, NIHR and the Wellcome Trust. She led the IID2 study and is leading the IID3 study. With that, I'll hand over to our wonderful speaker to discuss her work on the IID3 study.
Thank you very much, Liam, and thank you to the Food for Thought team for inviting me to give an update on the IID3 study to date. And the first thing I should do is start off by thanking both the Food Standards Agency and Food Standards Scotland for funding this study. So I guess the first thing to ask is, why is it important? Well, essentially, the two previous studies and this one provide essential building blocks in understanding the burden of illness and burden of disease from infectious intestinal disease, and they reveal the hidden burden of illness. And on the right-hand side of the slide, you will see an iceberg. And what that tells you is that for every case of infectious intestinal disease reported to routine surveillance systems, the IID2 study uncovered the fact that there were an additional 147 cases in the community. And without that kind of calibration, you can see that routine surveillance would hugely underestimate the actual burden of illness. And it matters not just from the point of view of people being poorly, but also helping to understand the economic impact of IID. Because obviously, the more people who are ill, the more people have to take time off work or school as a result of IID. So calibrating national surveillance systems is a very important purpose for studies of burden of illness. The IID3 study is also a world first. There are other studies of this type in other parts of the world, notably the Dutch, who conducted a study similar to IID1. I don't think anybody has so far attempted to do this 3 times. So, as I say, it's a world first. So I guess the next question is, what does the Food Standards Agency want to know from this? And the main question is, has the incidence of IID in the community changed since the late 2000s, which is when IID2 was conducted? As part of that, we want to be able to look at the overall incidence of IID in the UK, to establish the quantity of disease, if you like, presenting to primary care, and clarify the burden that is UK acquired, as opposed to people who pick up the infection overseas as part of tourism or business. We want to describe the pathogens causing IID in the UK and also consider levels of antimicrobial resistance. We want to recalibrate UK surveillance data for infectious intestinal disease overall, but also by pathogen. And we want to look at the numbers of cases presenting in the community to general practice, being admitted to hospital, and in hopefully rare events, dying from IID. And we also want to compare the results of this current study with previous studies, IID1, which was conducted between 1993 and 1996, and IID2, which was conducted in 2008-9. So we're going to have a snapshot of disease. Well, we're going to have 3 time points over a period of about 30 years when IID3 concludes. So how do we propose to do it? And I think it's worthwhile spending a little bit of time on this slide so that it's clear what we're doing and why we're doing it. So the IID3 study actually comprises a number of studies within it. The first is a prospective cohort study, the so-called cohort 1, and in that we intend to measure illness in the population. And we're going to do that by recruiting households, volunteer households in the UK, and following them up over time to see who develops illness, and if they develop illness, to get a stool sample from them to find out what's causing their symptoms. So that is our prospective population-based household cohort study. And that is, if you like, the lowest, the bottom layer of what is at the moment an inverted pyramid. So top layer here on this diagram. Oops. The next thing we're going to do is look at disease presenting to primary care. And we're going to do this in 2 ways. First of all, we're going to collect from every person who presents to their primary care team with symptoms of diarrhea and vomiting. We are going to collect a sample from them. Now, this would not necessarily normally happen. There are various algorithms for dealing with IID in the community, and it's entirely possible that from the purposes of the clinical management of a case of IID, a GP would not necessarily feel they had to take a sample first. So in this instance, in cohort 2, the GP presentation study, we are interfering with clinical practice to say, do you know what, even if you wouldn't normally sample the case, please do so. We are then sending those samples to our laboratory, where we are going to test them for a very wide range of pathogens. And again, not normal pathogens which are not normally sought in routine stool sampling at the moment. Now, this gives us our first under ascertainment measurement. So we've got illness in the population, and we will be able to calculate an incidence rate. And then we've got illness presenting to primary care, where we will also be able to calculate an incidence rate. And we already know from IID2 and IID1 that the greatest potential loss of information about IID in the community comes between these 2 steps. So many people with diarrhea and vomiting won't bother to consult a GP, and those that do may not get to see the GP in time to present a stool sample. And we know from IID2 that for every case of IID presenting to primary care, there were an additional 23 in the community. So as you can see, there's a big potential loss of information between cohort 1 and cohort 2. Now, with the GP enumeration study, which is cohort 3, we will simply observe what happens in normal clinical practice. So we will look at What happens when a patient presents to the GP? Does the GP ask for a sample? And if the GP asks for a sample, what does a routine laboratory protocol turn up in that sample? So there will be usual clinical decision making about stool sampling, and in that instance, the samples will go through the normal local laboratory testing algorithms. That gives us our second level of under ascertainment in IID2. So the difference between sampling everybody who presents with diarrhea and then sampling according to normal clinical practice, if you like. Then we will audit recruitment to the GP presentation study. And this is because we know that in the business of a busy clinical consultation, someone who could be recruited to the IID3 study might not be. So what we will be doing here is examining, if you like, the efficiency of recruiting into cohort 2. So we will do a case note review in practice, looking at those people who could have been recruited into IID3, but for whatever reason weren't, either because the doctor or nurse didn't have time in the consultation, or because the patient said, actually, do you know, I don't want to do it. And that will give us another level of under ascertainment. The samples that are taken either in the presentation study or in the enumeration study will go for laboratory testing. As I've mentioned, in the GP presentation study, all the samples will be tested for a wide range of pathogens, and those tests are being performed in one laboratory in Liverpool, which is doing the microbiology work. And then in the enumeration study, the orange arm in this diagram, as I say, it will be the normal laboratory protocol conducted by the local lab for that particular general practice. And that gives us another level of under ascertainment in surveillance data, because we know that no pathogen detected, which is the routine side of the equation, if you like, is not the same as no pathogen detectable, which is where our non-selective testing is taking place in Liverpool. And then we work out the rates of disease in all these bits of the pyramid and compare those rates with what appears in national surveillance data, which we can recalibrate using this pyramid approach, and that is the national reporting study. So our sampling frame for the IID3 study is the University of Oxford Royal College of General Practitioners Research and Surveillance Centre, which comprises a nationally representative network of over 1,800 general practices across the United Kingdom. The easiest way for us to sample the population is via primary care, and that is what we are going to do. So we are inviting GP practices to take part in the study, and then the GPs in those practices will recruit participants for us. The case definition is the same as that which was used in IID1 and IID2, so we could argue about its sensitivity, specificity, etc., but essentially this has been gifted to us by the original teams. So cases are people with loose stools or clinically significant vomiting lasting less than 2 weeks in the absence of a known non-infectious cause, and this needs to be preceded by a symptom-free period of 3 weeks. And this just makes sure that we are not sampling the same episode of diarrhea and vomiting by having that 3-week symptom-free period. Vomiting is considered clinically significant if it occurs more than once in a 24-hour period, and if it incapacitates the case or is accompanied by other symptoms such as cramps or fever. And to think about clinically significant vomiting in a practical sense, think norovirus. It's the type of symptom that overtakes you very suddenly and very violently, that really, norovirus really encapsulates what is meant by clinically significant vomiting. In terms of the target organisms we're looking for in IID3, as I've mentioned, we're looking for a wide variety of pathogens. We're testing for more organisms in IID3 than we did in IID2. So in IID3, for example, we are looking for Aeromonas, Vibrio species and Entamoeba histolytica, none of which were tested for in IID2. The organism we're not looking for in IID3 that we did in IID2 is Listeria monocytogenes. And the reason for this is that there were no cases of Listeria monocytogenes in IID2, and there were certainly no reports of stool samples being positive in patients with listeriosis. As you know, Listeria is basically an invasive disease, so you're more likely to find Listeria in blood or cerebrospinal fluid samples than you are in stools. So we're not looking for LM this time round. Our primary diagnostics are PCR based, so non-culture methods. And I'll come on to our laboratory algorithms a little in greater detail a little bit later on in the presentation. But as you can see, there's a wide range of bacteria, protozoa and viruses that we are going to be looking for in IID3. So in terms of the pilot study, the objectives were to gauge interest in the study from general practitioners. As you will all be aware, both the structure of primary care has changed quite a lot in the decade or so since the IID2 study was done, not only in terms of the organisation of primary care into larger groups these days, but also in terms of the pressure on primary care teams. And so one question was, is this viable? Is it viable to try and do it this way? The second question was the second objective was to design, build and test an end-to-end secure electronic system for patient recruitment, sample submission and laboratory reporting. To develop an application to NHS England for supplementary data that we're going to need to use in calculating the burden of illness, including hospital episode statistics, outpatient data, emergency department data, and data from ONS on deaths. And then to establish and refine our study procedures relating to diagnostic and reference microbiology. The pilot study started on the 21st of March and finished on the 23rd of June. So in terms of gauging interest from GPs, we needed to make the offer, if you like, as attractive as possible. So first of all, we registered the study with the National Institute for Health Research Central Portfolio Management System. And an important reason for doing that is that it unlocks support from the NIHR Clinical Research Network, both in terms of cash, but also in terms of resources, human resources, if you like, to help get the study off the ground. We also registered the study with the NIHR CRN Associate Principal Investigator Scheme, and that gives recognition to the individuals in primary care who take on the study, and they get training, and they get recognition, which I think is very important in terms of trying to make a study like this work. During the pilot period, we had contact with 190 general practices. We received 60 expressions of interest, and 22 practice agreements were signed during that period, 5 for cohort 1 and 17 for cohort 2. Now, we had estimated that for the pilot study, we were going to need to recruit at least half a dozen practices, and as you can see, more came on board than were originally envisaged to be needed, which was good. It certainly made it appear that general practice was going to be keen to engage with the study. So the second thing that I said we wanted to do was to design, build and test an end-to-end secure electronic system for participant recruitment, sample mailing and processing, and getting results back into the patient notes. One of the things that came up in our conversations with GPs, and I think we knew it anyway, was that in order to make this study fly, we were going to have to mimic as far as humanly possible the process which takes place in a normal consultation in order to make it as easy as possible for GPs to recruit cases. So the idea of this is that when a patient consults with infectious intestinal disease, the general practitioner can call up a screen, an ordering screen in the practice. They can take consent from the patient whilst they're sitting in the chair in front of them, and they can put in the order for the IID3 suite of tests. This is the so-called ICE desktop system. The patient provides a sample, which is then sent to the local laboratory serving the general practice. That is identified in the local laboratory as an IID3 test and recorded in their laboratory information management system. The sample is then sent through the XLab Labnostic network to Liverpool, where the tests are performed, and the results flow back from Liverpool through the XLab system into the local trust's laboratory information management system, and thence back into the patient's notes. So one advantage of this is that there is a very strong audit trail all the way through the system. The second is that it is obviously designed within the NHS infrastructure that gives as secure as possible a route for information transfer directly back into the patient's notes, and the GP can then review what has been found in the IID3 study. The other flow of information from the Liverpool clinical laboratories, as well as that going back to the general practice, is into the trusted research environment at Oxford University, which is called ORCID, the Oxford Royal College of General Practitioners Clinical Informatics Digital Hub. And what that does is give us a pseudonymised research data set that can then be analysed by the IID3 study team to do all the things which I mentioned earlier on in the presentation, looking at rates of illness presenting in the population to primary care, looking at the rates by pathogen, and recalibrating national surveillance. And this is an accredited hub, which again is recognised as having very secure information governance criteria. So that are well recognised by the NHS and also by the health research regulatory people. Now, this slide gives you an overview of what needed to happen. The next slide shows you in detail all the steps that we had to go through in order to make that system work. So there was a lot of work required in primary care, in the lab, and then from the lab back to primary care and also to Oxford. So on the outer columns of the slide, you've got the interventions that were needed in the GP systems in order to make this system work, and in the middle you've got the laboratory workflow as well, and I'll come back to that in a minute. But as you can see, there was a lot of steps that needed to work in order to make this end-to-end system both work and viable. Okay, and it did work. It worked for half a dozen practices in the pilot and is now, as you can imagine, being rolled out for the main study. We completed the NHS, we completed the information requirements for the NHS Data Access Request Service application, and we agreed the IID10 codes to be used for hospital episode statistics, emergency department data, and data on deaths from ONS. As some of you may be aware, IID11 is sort of hovering around in the background, but that's not going to be implemented in the NHS until April 2026, by which time the data collection for IID3 will be over. So although it was raised by reviewers, should we be looking at ICD-11, unfortunately it's not going to be implemented in time. And in any event, because we're going to be comparing the results of IID3 with IID2 and IID1, then using ICD-11 wouldn't be particularly helpful. The fourth objective, as I mentioned, was to establish and refine our diagnostic and reference microbiology procedures. In the diagnostic lab in Liverpool, we are in the main using the SeroSep platform, and you can see the organisms for which we're using the SeroSep platform, so Campylobacter, Clostridium, the Salmonellas, etc., using the enteric bio panel, and for the viruses we're using the enteric bio viral panel 3. For those target organisms for which there was not a PCR test in the SeroSep panel, we are supplementing that with genetic signatures easy screen, which we're using for a smaller range of pathogens, mainly the diarrheogenic E. coli, Cyclospora and Aeromonas. And then we have some individual PCR tests as well for VTEC. And for enteroaggregative E. coli and for enterotoxigenic E. coli. So those tests were set up, established, validated in the Liverpool Clinical Laboratory, and as I'll show you a little bit later on, they do work. And then we had to establish the flow of samples and how we were going to use the samples for the various for the platform. So obviously the pot comes in from via the MPEG system from HazeDX. The sample is receipted in Liverpool and then sent either down the easy screen route or the enteric bio route, and then any PCR positive samples are sent on to the reference laboratory, which I'll come on to in a minute. We are also preserving an aliquot of stool sample in the Good Laboratory Practice lab in Liverpool to form a biobank of stool samples from this study. And there are, as you will be aware, there are banks of samples from the previous 2 studies as well. PCR positive bacterial stool samples that are PCR positive for bacterial pathogens are sent to the gastrointestinal bacterial reference unit in Colindale, and they are then cultured, and organisms are undergo whole genome sequencing using Illumina chemistry. So the reference laboratory will then send back those results to Liverpool for transport back to the GP. And of course, this also allows our colleagues at Colindale to look at any potential outbreaks that we might pick up in IID3. Public Health Wales is doing the reference work for Cryptosporidium, Giardia and Cyclospora. And again, those results will be fed back to Liverpool, and if an outbreak is identified, to health protection teams. And the data from Liverpool, Colindale and Public Health Wales will all feed into the 2nd generation surveillance system that is our routine surveillance data, if you like, that the 4 public health agencies collate. If you're interested to keep up to date with where we are, then there is a link to a study dashboard from the Food Standards Agency's website, their webpage about the IID3 study. As you can see from the diagram, or from the chart, the nice thing from our point of view is that the PCR-based methods are lighting up for a variety of pathogens. So we've had some Giardias, quite a few diarrheogenic E. coli's one way or another. There's been a crypto in there, some Campylobacters. I think the only thing that we haven't had so far is norovirus or rotavirus. So keep an eye on the dashboard, and that will show you how the pathogens are accumulating over time, if you like. And the map on the right-hand side just shows you where our IID3 study practices are located across England at the moment, but Scotland, Wales and Northern Ireland will be coming on board. In terms of, does anybody care? Well, I think they probably do. As part of another exercise entirely, we just had a look at the global impact of the IID1 and IID2 studies, and this is based on the peer-reviewed papers that have arisen from both studies. And out of 25 peer-reviewed papers, we've had over 2,500 citations globally, which is not bad going, actually. And the majority are in Europe and in, shall we say, higher income countries. But it has had a surprising, or those 2 studies have had a surprising reach. And these are citations not just in the peer-reviewed literature, but also in the grey literature, in government reports in other countries, citations by the World Health Organization, and even by the Prime Minister's office in Iceland. But I think my favourite was a citation in Uzbekistan, which was a surprise. So I think when the results of this study are available, people are going to be interested. We already know that the World Health Organization is going to be looking keenly at what we find, because they are just setting up the next round of their foodborne epidemiology reference group, which will be estimating the global burden of foodborne disease. So I think these results will be important not only in the UK, but also elsewhere. And I think that's it, apart from to thank the study partners and also thank again the Food Standards Agency and Food Standards Scotland for funding IID3. And that's it from me. And I'm happy to take any questions.